This is a production of Cornell University.
All right, our last presenter is Jeremy Pardo. He's from Westlake Hills, New York, and was advised by Karen Bowerly and Ed Buckler on this project. After he graduates in a couple of weeks, Jeremy's going to go on to a PhD program in plant biology at Michigan State, which is great. And the seminar title is Transcriptional Regulation of Drought-Induced Stomatal Closure in the Maize NAM Population. Say that 7 times. This is just a tiny bit of his data. We really had to work to keep him within. This is like a PhD project.
In 12 minutes, go.
Yes.
So thank you. As Mike said, my name is Jeremy, and I, for the last couple years, have been working on my honors thesis under the supervision of Ed Buckler and Karen Bowerly. And so what I really wanted to do is look at how the transcriptome is involved in regulating stomatal closure. See how this works. There we go. So we'll just use the computer because it works better. And so what I wanted to do is look at these three aspects of how a plant works. So the physiology, and then the transcriptomics, so mRNA, and then try and integrate that together with some genetic mapping to really get at how stomatal closure under drought is working. And so what is this stomatal closure that I'm talking about? So stomates are pores on leaves, and plants can close them in response to water stress to limit water loss. But by closing them, they also limit photosynthesis, so it's a trade-off. And the plant hormone abscisic acid is very much involved in mediating this response. However, that hormone is also really important in controlling other genetic responses to drought. And I should mention that there are drought response mechanisms that are independent of abscisic acid. For example, the hormone ethylene plays a major role too. But for today, I'm going to focus on abscisic acid. And so the plant material that I used for my experiments was taken from the maize nested association mapping population. And the lines that you see up on your screen are the founders of this population. So there's 27 inbred corn lines from around the world that sort of represent the diversity of maize as best as you can with 27 lines. And then you see the two that are highlighted in red were the parents for the mapping population for that portion of my experiment. And so I conducted two separate experiments. One I'll refer to as the quantitative trait mapping experiment, and the other as the RNA sequencing experiment. And for both of them, I grew plants for 3 weeks, and then I took the seedlings and I started to drought stress them. And so for the quantitative trait mapping experiment, on what I called day 0, so I had 3-week-old plants. They were well watered, and I started measuring some physiological traits. And then by day 1, I stopped watering them, and I kept measuring those traits each day. And finally, on day 4 of that experiment, I re-watered them and then measured again to see how the plants responded. And for the RNA sequencing experiment, it was a very similar design, but here I had 2 groups. So the control group was always well watered, and the drought stress group, very similar. So day 0, they're well watered plants, and by day 1, I've withheld water and they're starting to become drought stressed. And on day 1 and day 3 of that experiment, I collected leaf tissue to do RNA sequencing. And so those physiological measurements, here's just an example of some of the things I measured. So the white little clamp device is called a pyrometer, and it measures stomatal conductance. So it's basically measuring the flow rate through the stomates, which is a measure of how open they are. And so the more open the stomates, in general, the less drought stressed the plants are. And then the other device on there with the little prongs is not a torture implement. It's a soil electrical conductivity meter. And then so I use that to measure the water content of the soil to get an idea of how drought stressed my plants actually were. And then for RNA sequencing, I used a method that was adapted to plants by Carl Kremling to do 3' RNA sequencing. And basically all you need to know about that is it's a way to cheaply and quickly profile the RNA from a whole bunch of samples. Yeah. Okay, so back to this physiology, mRNA, and genetic mapping. So first I'm going to discuss the physiological results that I got. And so this graph is showing the stomatal conductance of those NAM parents. And the red boxes are the control group, so those were always well watered. And the blue boxes are my drought stress treatment, so I withheld water from that. And what you can see is by day 1 of my experiment, there's a significant decrease in stomatal conductance in the drought stress group. But you might also see that there's still a lot of variation on day 1 between genotypes. And so this plot is very similar. It's just showing the data, the same trait, the stomatal conductance, but in my quantitative trait mapping experiment. And again, by day 1 of the experiment, there's a significant decrease in stomatal conductance, but there is also still a lot of variation between genotypes. And to look a little bit at that variation between genotypes, so this plot is showing the ratio of stomatal conductance on day 1 to day 0 for all of those lines in my RNA sequencing experiment. I think the battery on this is dead, so the pointer won't work. But what you can see is that, yes, there's a lot of variation within genotypes, but also between genotypes. So the red line represents a ratio of 1, so that would mean that there was no change between day 0 and day 1. And anything below that line means that the stomatal conductance decreased. Anything above that line would mean that it increased. And you see that basically all of the genotypes are decreasing their stomatal conductance, but some of them are right around that red line, whereas some of them have decreased dramatically already by day 1. And so to sum up my physiological results, I found that stomatal conductance decreases significantly under drought stress, but there is a lot of variation between genotypes. And so to sort of get at what that variation means, I wanted to look at the transcriptional control of this. So I started wading into my RNA-seq data. So what this plot is showing is that it's a principal component analysis. And so what that is a way of reducing very high dimensional data down into 2 dimensions, so you can put it on a graph and sort of make sense of it. And so all of the points in purple are the day 3 drought conditions. In green are the day 1 drought conditions. And then red and blue are the control samples. And what you can see is that on day 3, there's sort of a cluster all the way over on this side of the graph, whereas the controls are on the opposite side, and the day 1 samples, their expression is somewhere in between. And so basically, the gene expression of my samples is clustering by treatment. And that kind of leads me to believe that the treatment is having a major effect on that gene expression. And so when I look at genes that were differentially expressed between the control group and the drought stress group on day 1 of the experiment, what I found is a lot of enrichment for genes that are involved in stress tolerance. So this makes sense. It seems that the plants are responding to drought stress, which is what I was hoping. And then to go from sort of general overviews of the expression to highly specific one gene, And so this plot is showing the expression of the rate-limiting enzyme in abscisic acid biosynthesis. And what you can see is that on both days during drought stress, it is significantly more expressed than under control conditions. And so that suggests to me that if the plant is upregulating the expression of an ABA biosynthesis enzyme, that is probably producing more abscisic acid. And so, as I had mentioned earlier, abscisic acid is really important in stomatal closure. And it works through this complicated signal transduction pathway that ends in stomatal closure at the end of it. But what's interesting, highlighted in that red box, is a protein phosphatase. And what that does is it negatively regulates the abscisic acid signaling pathway. And so this is the expression of just one of those protein phosphatase genes. It's actually a whole family, and I found that 9 of them were differentially expressed under drought. And this is just one of them, which has already been shown to be important negative regulator of that pathway. And interestingly, it's also expressed more under drought. So the plant is making more abscisic acid, but it's also making more of a negative regulator of the abscisic acid signaling pathway. And so I wanted to know, like, what is coordinating this delicate balance between the negative regulation of this pathway and the upregulation of the pathway. And so the answer, I think, lies in transcription factors. And so I looked at all of the transcription factors that were differentially expressed under drought. And I found that there were 500 different transcription factors that were differentially expressed on at least one of the days under drought stress. And what I did is correlate their expression pattern with the stomatal conductance. And I found that 30 of them are significantly associated with stomatal conductance. And then among those, I had enrichment for these 2 general families of transcription factors. So the heat shock transcription factor family and the BZIP transcription factor family. And so heat shock transcription factors are known to be involved in a stress response. And the BZIP family is one of the largest transcription factor families in plants, but there's a subset of them that are very important in regulating abscisic acid-related processes. So I thought I'd look at those a little more. And so this is just one example of one of the BZIP transcription factors that was differentially expressed under drought. And you can see that on both day 1 and on day 3, there's much higher expression. And so this BZIP 68 is regulated by abscisic acid. And what it does is it controls chlorophyll degradation. And how is that related to drought? Well, under drought stress, as I mentioned, the stomates close, and that limits photosynthesis, but it doesn't affect how light is absorbed by chlorophyll, and that results in damage. And so one of the responses that the plant has is to degrade some chlorophyll to limit the damage. Okay, so that was really interesting, hopefully, but I wanted to integrate that a little more with what is controlling stomatal conductance. And so I looked at my genetic mapping data, and if you remember, that was a separate experiment using a population that was derived from 2 of those parents where I had RNA sequencing data for. And so I looked on both day 1 and day 3 and tried to correlate stomatal conductance with genetic markers. And so this is a Manhattan plot, and each dot represents a SNP marker. And so the higher the dots are above the line, the more associated they are with stomatal conductance. And what I found is that I had a significant region of the genome on chromosome 8 that was significantly associated with stomatal conductance on day 1. And then when I looked at day 3, I found something similar. Again, I had one really peak on there that stands out, and this time it was on chromosome 2. And so now that I have these transcriptional data and the physiological data, as well as this genetic mapping data, I wanted to look at how this all comes together. So the intersection of all of these aspects of the plant's growth and physiology. And so what I did is look in my RNA sequencing data, which, if you remember, is a separate experiment. But I looked in the regions that were significantly associated with stomatal conductance from the genetic mapping experiment. And so on day 1, I did find that there were genes differentially expressed, but unfortunately they're uncharacterized proteins, so we have no idea what they do, and so I'm not going to talk about them any further today. Feel free to ask me about them later. But on day 3, there were 2 genes within that chromosomal region on chromosome 2 that was significantly associated with stomatal conductance that were also differentially expressed under drought. And so one of them in particular is this HVA22-like protein. And so what that is, if you can see, it is differentially expressed under drought, and it's expressed more under drought stress. But if you look at those little red and blue dots, which are the parents of that mapping population, it's kind of hard to see, but under control conditions, they're at sort of opposite ends of the expression spectrum. And they don't seem to be at opposite ends under drought stress, but at least under control conditions, there seems to be a big difference between my 2 parents, and this gene is differentially expressed under drought. And so what it is in barley, this HVA22 is a programmed cell death inhibitor that is responsive to the hormone abscisic acid. And so the way seed germination in barley works is there's a layer of the seed called the aileron layer. And when the seed germinates, there's programmed cell death, and a bunch of enzymes are released that degrade the seed coat. And you see unfortunately this pointer's not working, but if you look at the middle box on the bottom there, you see that you have these circles with nothing in them, because basically the cell has died and all of the goop has been released. But when you have overexpression of the HVA22 protein, you can see that the cell is not dead and it looks the same as the control conditions. And so this gene is inhibiting programmed cell death. And so I am hypothesizing that in leaves, this HVA22-like protein might inhibit programmed cell death that's involved in senescence, which we know is triggered in part by drought stress. And so just to sum up what I've told you so far, the key ABA biosynthesis gene is differentially expressed under drought, and it's upregulated. But so are negative regulators of the abscisic acid signaling pathway. And my best candidate for the chromosomal region that was associated with stomatal conductance is an abscisic acid responsive protein that seems to be an inhibitor of programmed cell death. And so in the future, what I'm going to do this summer is look for common motifs in the promoters of all of the genes that I found that were differentially expressed under drought, and hope to find like these sequences that are associated with a binding of transcription factors that maybe are controlling drought tolerance. And so finally, I would like to thank everybody who made this project possible, so especially my 2 advisors, Ed Buckler and Taryn Bowerly.
Questions for Jeremy?
Yes.
Jeremy, in experiments where a dry down cycle has been done in a limited pot volume, sometimes what can happen is that lines that just generally are runts that have very low rates of photosynthesis and low stomatal conductances will remain well hydrated even after 3 days, while the ones that are really healthy and are transpiring rapidly deplete the water very quickly. And then you can get some, it's hard to interpret data like that. I was wondering whether you feel that your system was a set of lines that didn't have those kinds of variations in the control transfer.
So there's definitely that variation, and so what I did to control for that, at least for the genetic mapping experiment, I included, I measured plant height and leaf number, and then sort of merged that together into a leaf area like factor, and I included that in the model when I was doing the genetic mapping. And so that was my attempt to control for that. However, it is still a major problem, and if I were to do the experiment all over again, I would probably use a larger pot volume, simply because that would help to mitigate that factor.
I wonder about the expression that PPC and ADA biosynthetic enzyme. So the expression of these two kinds of sort of opposite function genes has been upregulated, right? And I wonder whether you measured them in day 1 and day 2, whether you can see by the time. So maybe, you know, ADA expression enzymes are up day 1, but PPC is up by day 3, or it's just involves two.
So they're actually both upregulated by day 1, and so I only had 2 time points. Maybe if I had measured expression every hour, there could have been like more differences. But the way, I didn't go into it, but the way that ABA works is it actually inactivates the PP2C inhibitor. And so one reason that this might be occurring is it's upregulating the negative regulator so that as soon as the drought stress goes away, the response can be turned off, and it might be more of a drought recovery mechanism. But that's just a guess.
Any questions? Okay, let's thank Jeremy again.
This has been a production of Cornell University, on the web at cornell.edu.